Hello friends, again welcome to my channel. Today we are going to discuss about the Arabinose operon. Now preferentially we know that operon it is a gene regulatory system which is responsible to control the expression of gene, that is the synthesis of different proteins which are required for the different metabolic activities. Now the Arabinose operon it is type of a biosynthetic operon, that is through it is a degradative operon, not the biosynthetic operon, because majorly through the activation of the genes involved within the arabinose operon, there is the degradation of the arabinose, which is an carbohydrate source for the purpose of energy requirement of the cells. Now, the arabinose operon can be defined the system carrying the number of genes like the regulatory, promoter, operator, inducer, and the structural genes which are responsible for the regulation of arabinose operon for the L-arabinose breakdown into the xylulose 5-phosphate. Now the different structural genes which are involved within the arabinose operon includes the gene B, A and D. So this arabinose operon it is also known as Arab-BAD operon. Now the Arab B, Arab A and the Arab D, these are responsible to transcribe the mRNA by encoding the kinase, isomerase and epimerase enzyme, that is Arab B responsible to synthesize kinase enzyme, Arab A for the isomerase and the Arab D for the epimerase enzyme. These are responsible to catalyze the conversion of L-arabinose to the xylulose phosphate, which is then enters into the pentose phosphate pathway. Now here it is a diagram to observe the construction of the arabinose operon, within which here we can see that there is the presence of the RSC gene. This RSC gene it is an regulatory gene which is responsible to control the expression of genes for the from the arabinose operon. Now here we can also observe there are the two different types of the operons these are present, or the operator genes these are present, that is the area O2 and the area O1, and these are the promoter gene for the area the PC, it is the promoter gene for the RSC protein. Then again there is the cap site, that is the catabolite activator protein site for the positive regulation of arabinose operon. Then the inducer gene it is present. Then the promoter for the expression of structural genes of the arabinose operon, that is the Arab B, A and D, and the Arab B, A and D, these are the structural genes. So it is the complete construction of the arabinose operon. Now majorly these three structural genes which are the part of this arabinose operon, the main function of these it is to encoding the metabolite enzymes which are essential for the degradation of L-arabinose into the xylulose 5-phosphate. The inducer gene, that is the area I, it is acts as an inducer gene and responsible for the induction of the transcription or the stimulation of the transcription process, that is there is the expression of these genes. The arabinose operon, the inducer molecule it is the arabinose, that is the directly the arabinose sugar, it is acts as an inducer within this arabinose operon like that of the within the lactose operon, which binds with the repressor protein to induce the transcription of the gene into the mRNA. That means here it is responsible to break down the repressor protein which is responsible to inhibit the process of transcription. Then the role of the catabolic activator site, that is the cap site. This activator site it is termed as the cap. A term cap stands for the catabolite activator protein which activates the efficiency of transcription rate. That means here there is the positive regulation of the arabinose operon. Majorly whenever there is the availability of glucose it is high with the low arabinose, the ATP will not convert into the CAMP. If the availability of glucose it is low with the high arabinose content, then the ATP will convert into the CAMP and the whole complex will then bind to the cap site to activate the transcription of mRNA. Means this catabolite activator protein it positively activates this arabinose operon whenever there is the low concentration of glucose and the high concentration of the arabinose it is present, because at that time there is the higher concentration of CAMP it is present which is responsible to activate this cap site. Then the operator genes. Now there are the two operator genes, that is the O1 and O2. Within the positive regulation whenever the amount of glucose it is low and the amount of arabinose it is high, the repressor protein, that is the RSC protein, will get activated by the arabinose. it will promote the synthesis of RABAD mRNA. And in the negative regulation, whenever the amount of glucose it is high, the amount of arabinose it is low, the repressor protein RSC will promote the RABAD operon. It is responsible to repress this Arabinose operon synthesis. Means majorly this RSC protein it is responsible for the both positive and the negative regulation of the Arabinose operon. In the presence of Arabinose, this RSC protein it is responsible as a activator protein, and in the absence of the Arabinose it will act as a repressor protein. Now the promoter genes. There are the two promoter genes in the Arabinose operon, that is the PBAD and the PC. That is PBAD, it is for the promoter site for the RABAD, that is the structural genes, and the PC it is the promoter site for RSC regulatory gene promoting the RSC repressor protein synthesis. The RSC repressor exists in the active P1 state in the absence of inducer, that is in the absence of Arabinose, because we know that this RSC it is acts as both activator and the repressor. So the active P site it is a repressor, it is exists in the active P1 state, and whenever it is inactive the P2 state in the presence of inducer. Means whenever it is present within the active P1 state it is acts as a repressor, and whenever in the P2 state it is in the inducer state. Now the regulatory gene, that is the RSC. This RSC it is the only regulatory gene within the Arabinose operon. This RSC protein regulates the Arabinose operon both positively and negatively. Whenever it binds with the operator, it will repress the synthesis of RABAD, and it will promote the binding of RNA. It will not promote the binding of RNA polymerase with the promoter region. In the activation of the RSC with the Arabinose, it will promote the attachment of RNA polymerase to the promoter region and leads to the synthesis of RABAD mRNA. The regulation of Arabinose operon. Majorly the Arabinose operon it is regulated both positively and negatively like that of the lactose operon. Now positive regulation it means there is the enhancement of the transcription of the structural genes. Now whenever both inducer and repressor protein it is absent, that is here means in the case 1 of Arabinose operon, if both RSC and the Arabinose these are absent, so in that case there will be no repression of the Arabinose operon in the absence of both inducer and the repressor protein, because majorly no doubt as the repressor protein it is absent, there can be the expression of the structural genes, but as the inducer it is absent, so due to the absence of inducer majorly there is no activation of the structural genes. Now the case second, it is whenever both inducer and the repressors these are present. Now we know that the Arabinose it is an inducer within the Arabinose operon. It will bind with the repressor protein to regulate the mRNA transcription. Now the RSC protein and the Arabinose these are responsible to form a complex that will not allow the loop formation, which is responsible to inhibit the binding of RNA polymerase. The Arabinose binds with the RSC dimer and changes its structural configuration. This change within the structural configuration of RSC it will allow the RNA polymerase to transcribe the RABAD genes to form the mRNA. Now here within this diagram we can observe such a condition, and due to the presence of inducer there is also the presence of the CAMP which is responsible to activate the cap site and which is also positively regulate the gene expression. Now the case 3 it is the when only repressor protein it is present. Now the main role of Arabinose operon involves the breakdown of Arabinose. In the absence of Arabinose there will be no transcription and the translation of the DNA molecule or this operon. The Arabinose acts as an inducer which binds and inactivates the repressor protein. As here we can say that only there is the presence of RSC and there is absence of inducer. The repressor protein will be produced by the RSC gene, and this RSC repressor protein forms a dimer. Here within this diagram we can observe that how there is the formation of this loop formation due to the presence of this dimer of the RSC protein with the operator and the inducer gene by forming a loop. Means here within that this O2 and the O1 both are get blocked, and majorly due to the RSC protein. The one RSC protein it binds with the array O1, that is the operator O1, and another bit is the array O2 and the inducer site. So due to that, majorly it forms a loop within the DNA which blocks the binding of RNA polymerase enzyme. The loop formation will not allow the RNA polymerase to transcribe the structural genes. Therefore, this Arabinose operon can be both positively and negatively regulated. And within simple words, we can say that it is a system which can be controlled by the activation and repressor with the RSC protein. The repression of Arabinose operon, it is through the repressor protein, that is the RSC protein, and the activation of the operon, it is through the inducer, that is the Arabinose. So we can conclude that the Arabinose operon, it is switched off in the presence of repressor and it is switched on in the presence of inducer, that is the Arabinose. So in short, it is the information about the Arabinose operon. So if you like this video, please subscribe my channel and also share this video with your friends. Thank you.